AP Bio. I'm going to be going over day 4. I'm going to start out by giving you some AP exam tips. This is becoming a daily thing, as you probably noticed, which makes sense because we're about a month away from the exam. Then we are going to have a little mini lecture about Unit 5. Then in the next video, there's going to be a mini lecture for Unit 6. That's going to be really short. And then I'm going to describe the homework for today. The review tips. First of all, same 2 lessons that I'm spotlighting today from College Board. The photosynthesis one is airing today, Thursday, and then the cellular respiration one is airing tomorrow, Friday. So I would recommend checking those out. And then another one I'm going to recommend is from the AP Bio teacher community. This lesson that's linked right here has content from our Unit 1 and 3. So it'll be about cell cycle, it'll be about cell communication, cell signaling, all of those sorts of topics. So I recommend checking that out. And then here, I'm just putting one more time the Zoom FAQ session that I ran about questions from the AP about the changes to the AP exam. Here's my working calendar, and here is this Something I Miss About You link, which, if you need a little smile, might be a good pick-me-up. It certainly is a nice little pick-me-up for me. Okay, next, just a reminder of what I'm talking about when I'm talking about the units. So today I'm going to be focusing on molecular genetics. College Board calls that gene expression and regulation, and then also evolution, which College Board calls that unit natural selection. Okay, so this would be a good time to go and get yourself a lined piece of paper and something to write with and pause the video. And then once you've got that, you can restart the video. So, a little mini lecture on Unit 5, Molecular Genetics. So we started this unit by talking about DNA structure. And if you remember, you did the little puzzle where I had the different color laminated puzzle pieces, and you were trying to fit them together and figure out, make the structure of DNA. And if you remember, DNA is a nucleic acid. It's one of the 4 types of macromolecules, and the building blocks for nucleic acids are nucleotides. And a nucleotide is made up of a sugar, a phosphate group, and a nitrogenous base. In the case of DNA, the sugar is called deoxyribose. That's why it's DNA, because DNA stands for deoxyribonucleic acid. And then the nitrogenous base can be either adenine, thymine, cytosine, or guanine. Adenine binds with thymine, cytosine binds with guanine. And then one of the next things we did in this unit was we did the activity where you had the marshmallows and the little Starburst chews, and you were using those to make to copy strands of DNA. And what we the conclusion we were supposed to come to with that lesson was that DNA replicates through a semi-conservative method, meaning that it's not that you have an old piece of DNA and then an entirely new piece of DNA. Instead, that old piece of DNA splits in half, and then you have, of your 2 new pieces of DNA, half is old and half is new. So it's semi-conserving the old DNA. And that's what we see here as well. So this top right here and this bottom here are both the old DNA, and then we have our new piece of DNA being added on. And there are lots of different enzymes that are involved in that, DNA polymerase being the most important. The other key thing we talked about was that because of the directionality of the way DNA polymerase works, there are these Okazaki fragments that are formed, so it is not a continuous line. And I would recommend reviewing more about that, but I'm not going to do that in this video to make sure that I'm keeping concise here. So then we talked about transcription, and I would strongly recommend drilling yourself a little bit on the differences and similarities between DNA replication and transcription, because those are common areas of confusion. Transcription is where we are copying DNA like DNA replication, except this copy of DNA is being turned into RNA. We're not just taking DNA and making an exact copy of it. We're taking DNA and copying that into RNA. And we're doing that with the goal of making a protein. Just like DNA replication, this happens in the nucleus. Then we have RNA processing. So remember, the RNA after transcription is not yet ready to leave the nucleus. There are actually some changes that need to be made to it. Specifically, the introns need to be spliced out or removed. The poly A tail needs to be added. The 5' cap needs to be added. So we need to do these things to protect the RNA before it leaves the nucleus. And also to make sure that we have the exact right code we're looking for. This image is helpful just because it has it sort of lays out for you the differences in structure between DNA and RNA. So I would recommend reviewing that image. And also this link here is the source of this image, and it does a good job going through that for you. So then the RNA leaves the nucleus, it's done with the processing, it leaves the nucleus, and it goes to a ribosome. Ribosomes are floating around in the cytoplasm. That's the sort of fluid that all of the organelles are floating in outside of the nucleus. And so the RNA goes to that ribosome. I guess the ribosome could also, of course, be on the rough ER if it's not just floating in the cytoplasm. So then the RNA comes here. Here we see the RNA. Specifically, we're talking about mRNA. It comes to the ribosome, and it binds up with tRNAs. And the tRNAs, each of these is a tRNA. Those have triplets of nucleotides at the end of them. Or of, nitrogenous bases. And these triplets of nitrogenous bases bind up with the triplets. They match up with the ones on the mRNA here, and that determines what amino acids are being linked together. And we also have this really great chart here called the Universal Genetic Code Chart, which tells you how this matching works. So for example, if we look for UUU, right there, U, and then pull over U. So we see UUU. Oh, and this is the UUU that would be on the mRNA. That would correspond to the amino acid that starts with PHE. Now, this UUU is because it's on the tRNA, so we would look for AAA. Remember that A binds with U in RNA. So AAA, go A, right here, corresponds, as we see here, with the amino acid that starts with LYS. So that's how you read this chart, and sometimes on tests you are asked to tell me what are the amino acids that are being coded for by this mRNA strand, and so you have to use this chart in order to figure that out. I would strongly recommend watching this video because it has a really helpful visual layout of the process from transcription through the end of translation. And I think actually it's pretty impossible to do this process justice just through talking through it. I think a video is pretty necessary to have the animations. So I would recommend watching that video. It's very short. I think it's 2 minutes or something like that. Okay, after you've watched the video, come back here, and then we're going to talk about genetic transformation. So, genetic transformation, what is that? So, our lab where we got the bacteria to glow was our genetic transformation lab. We also called it sometimes the bacterial transformation lab. Same thing, just specifically in bacteria. Genetic transformation is where you are changing the DNA of an organism with the goal of changing that organism's trait. The way we were changing the DNA of our organism, E. coli, was by adding a plasmid to it. You guys added 2 different versions of plasmids. A plasmid, remember, is a small circular piece of DNA, and bacteria naturally have these plasmids, so it's pretty easy to insert a plasmid into a bacteria cell and get that bacteria cell to start doing transcription and translation to use that plasmid to make new proteins. And our goal was, if you remember the plasmid we had, it wasn't exactly this one, but it's very similar to this one, where it had a gene for glowing. And so when we inserted that plasmid into the bacteria, it would start making the glowing protein, which would make it glow. But remember that there was a catch. There were in fact 2 catches. One was that we had 2 different types of plasmids. One of them, if you got the bacteria into, or if you got the DNA into the bacteria, it should be able to make it glow right away. The other one only glowed when there was a rhabinose present. And the reason for that is because there was a little bit of gene regulation going on, where the gene for glowing was only turned on when there was a rhabinose there. So if there wasn't a rhabinose, that gene wouldn't be turned on, which meant that the glowing protein wouldn't be created. Now, the other thing going on was we had some antibiotics in the mix. So we had added ampicillin to some of the plates. And the reason for this, if you remember, was that all of our plasmids had a gene for ampicillin resistance. And that was important because if we didn't select for only the bacteria that had ampicillin resistance, then the normal bacteria that wasn't able to glow outcompeted our bacteria that were able to glow, and we didn't actually get to see any glowing. So for example, if we had a plate that had a rhabinose, we hadn't put the plasmid, we had to put the plasmid in the bacteria, and you'd think now the bacteria is going to glow, but we still wouldn't see any glowing. We'd see plenty of bacterial growth, but we wouldn't see any glowing because we hadn't selected for only the bacteria that had the plasmid. Okay, and then that is related to this free response question. So I'm going to encourage you to pause the video here, read over this question, and then try answering it, and then I'm going to go over it. Okay, so hopefully you gave this an attempt. I'm going to go over this question. We're just going to go through the key because I think this is a good way to do both some free response practice and do a little review. So this question is asking you about a very similar topic. We have some bacteria. That bacteria we're inserting plasmid into, and this plasmid has 2 genes that code for antibiotic resistance, but for 2 different types of antibiotics, kanamycin and tetracycline. So first, it says, what do you think would happen if you have transformed E. coli in the presence of tetracycline? Transformed means that you have correctly added this plasmid into the bacteria cell. So if you have correctly added the plasmid into the E. coli bacteria cells, and it's in the presence of this antibiotic, which normally would kill the bacteria, but since you've added this plasmid correctly into the E. coli, because it says transformed, then the bacteria should be able to grow. The bacteria population will increase. And this is because, so our justification is that this is because the bacteria is going to express the TET resistance gene, meaning express the gene that makes it resistant to tetracycline. Or you could say it's because the bacteria is making the TET resistance protein, meaning it is making the protein that makes it resistant to tetracycline. Gosh, this is hard to say. One thing I will say is that I often have seen when I give this question to students that they'll say it's because the bacteria has the TET gene, and that actually wouldn't have gotten you the point here. You have to specifically say that it is expressing the TET resistance gene, or it is making or synthesizing the TET resistance protein. Seems like a small difference, but that could be the difference between a point and no point. Okay, the next part of this question says that the researchers put this human DNA in, and now all of a sudden the bacteria is not able to survive in the presence of kanamycin. So you had to draw the X somewhere on the spot here for the kanamycin resistance gene. And your justification is that for some reason, that gene is disrupted. So the human DNA disrupted the CAN gene, or the human DNA disrupted the CAN promoter, like if you did it specifically over the dark area. Or these bacteria cells must not have an intact or correct or functional CAN gene. All of those would be correct. Okay, hope you did well on that. So that is it for this video. The next video, I'm going to be doing a little mini lecture on Unit 6. Okay, thanks guys.